Bismillahirrahmanirrahim. Assalamu alaikum dear students, I hope you all are fine. So in continuation with the online lectures for the course of medicinal chemistry, so today the first lecture will be on the biomolecules. So we'll consider the key concepts, and among key concepts we'll consider what do we mean by these biomolecules, what are carbohydrates, what are lipids, proteins, nucleic acids and enzymes. So let's start with the organic biomolecules, right? So chemicals or molecules which are present in the living organisms are called biomolecules. And another term that you should know that the different type of biomolecules, some of all type of biomolecules present in a cell, right? So that includes the different type of biomolecules themselves, the compounds, or the ions present. So all of these, when present in a cell, that's called the cellular pool, right? While these biomolecules, what are they? They are chemical or molecules. These are the organic chemicals or organic molecules which are present in the living system, right? So in this context, we are considering the different biomolecules, and then this specific chapter will consider about the organic biomolecules. So organic biomolecules are the compound of carbon, and you know that carbon is unique because from the general basic biochemistry, you know. So from the general organic chemistry, you know that carbon is unique because this carbon has 4 valence electrons, can form 4 bonds, and it can form 4 bonds with the same type of atoms or the different type of atoms. Furthermore, this carbon can form a single bond, a double bond, a triple bond with itself or with other elements, and can form rings or it can form chains, right? You can see or you can imagine that how much diverse a carbon is, right? So the organic biomolecules, these are compounds of carbon. So there are basically 5 major classes of biomolecules, and we are considering here the biomolecules which are macromolecules, right? So what do we mean by those macromolecules? We'll consider shortly, but you should know that there are major organic macromolecules. They have 5 classes. So these include these carbohydrates, lipids, proteins, nucleic acids and enzymes. So we will describe them one by one, but we will remain brief on these topics. So firstly, the carbohydrates. So carbohydrates are important for us because they are the main source of energy for all living organisms. It is also important for you to note that carbohydrates are not only the main source of energy for all living organisms, but these are also the immediate source of energy for all living organisms, right? So whenever you eat something and you get immediate energy, that immediate energy you are getting, this is because of these carbohydrates, right? So these carbohydrates are composed of carbon, hydrogen and oxygen atoms mainly, and these carbon, hydrogen and oxygen atoms are found in 1:2:1, right? So they should be 1 ratio of carbon, 2 ratio of hydrogen and 1 ratio of oxygen, right? So whenever you take a carbohydrate, so when this carbohydrate is broken down, it is broken down as sugar for cells, right? So this sugar which is broken down from this carbohydrate, so this sugar is used to carry out cellular activities such as the cellular respiration and photosynthesis, right? So you should know about the breakdown products. The breakdown products of these carbohydrates are the sugars, right? So carbohydrates are basically macromolecules. So what do we mean by macromolecules? Macromolecules are large molecules which are basically polymers of their monomers, right? So for example, this carbohydrate is a large size molecule because that's a polymer, and its polymer and the monomers of this carbohydrates are called the monosaccharide, right? So the carbohydrate is a macromolecule. It's a biomolecule, and this carbohydrate is basically a monomer, is a polymer, and its monomer are known as the monosaccharides. Right, so what do we mean by sugar? Right, so sugar is basically composed, simple sugar is basically composed of one or two monosaccharides. Right, so what are monosaccharides? Monosaccharides are basically the monomers which form the carbohydrate. Right, and the simple sugars are composed of one or two monosaccharides. Right, so examples of simple sugars include glucose and lactose and fructose. So all of these are examples of these simple sugars. Right, so carbohydrates can be found in sugar. You know, sugar is basically a sucrose. Right, the table sugar is a sucrose, and sucrose has two monosaccharides in it. Similarly, milk is also a disaccharide. It has two monosaccharide units, and these are fruits. So each fruit has carbohydrates, and it has large number of monosaccharides in it. Right, so what are the polysaccharides? So we can define them as, so when three or more monosaccharides combine, they form what's called a polysaccharide. So polysaccharides are also known as the complex sugars. Right, so monosaccharide and disaccharide, they are known as simple sugars, while the polysaccharides are known as complex sugars. Right, so these polysaccharides are also known as glycans. For example, polysaccharides, examples of polysaccharides include the bread, the rice, the pasta, and the potatoes, etc. So all of these are basically the polysaccharides. Right, so some common polysaccharides we can show here. So most animals store their excess sugar as glycogen. Right, so this glycogen is used when it is needed by the muscles or when the blood sugar is low. Right, so your body has the ability to store the extra sugar, and this extra sugar is stored in the form of a glycogen. Right, and whenever we need, for example, if you are starving and you need energy, then your body starts consuming this glycogen. Also, when you contract your muscles, then in that case glycogen is also used as a source of energy. So this glycogen is basically a polysaccharide. So just like animals store their energy, their sugar, in the form of glycogen, the plants store the excess of sugar as starch. A third example of polysaccharide is cellulose. Right, cellulose is also a polysaccharide. It is found in plants, and the rigidity as well as flexibility of plants is because of this cellulose. You can see that the plants can bend easily, but they don't break easily. Right, so they are rigid, but they are also flexible. So the cellulose is actually the polysaccharide which is responsible for the rigidity as well as flexibility of plants. So these three are the examples of some polysaccharides. So you can find some more examples of polysaccharide as well. So here we have considered three examples: the glycogen, the starch, and the cellulose. So just for review, the carbohydrates are the immediate source of energy, right? And they have carbon, hydrogen, and oxygen in a ratio of 1:2:1, right? So carbohydrates are also known as the hydrates of carbon. And why they are called hydrates of carbon? Because in carbohydrates, the ratio of hydrogen and oxygen is same as found in water. Right? So I mentioned that it has carbon, hydrogen, and oxygen, and the ratio is 1:2:1. So it has 2 hydrogen and 1 oxygen. You can get that from this. So because of the presence of a ratio of hydrogen and oxygen same as the ratio that's found in water, therefore the carbohydrate is also known as hydrates of carbon. Right? Further, we saw the carbohydrates are basically polymers, therefore we consider carbohydrates as the macromolecules. And the polymers, these polymers are made up of the monosaccharides, and the monosaccharides are actually the building blocks of these macromolecules. Right? So further we saw that the monosaccharides, which are also known as simple sugars, so when we have 2 simple sugars bound together, we form a disaccharide. So the example of a disaccharide is common sugar or the table sugar, which we use in our homes. So You may be asked by someone that what is the table sugar which we are using in our home. So it's not glucose, it's basically the sucrose, right? So this sucrose is basically a disaccharide. Then we saw that what are polysaccharides. Polysaccharides are basically when more than two or more than three monosaccharides combined together, then they form a polysaccharide. So polysaccharides are also known as complex sugars, and they are also known as glycans, right? Then we saw some common examples of some polysaccharides such as glycogen, starch, and cellulose. So now we'll consider another example of these macromolecules, and here we will study the examples of lipids. So lipids includes the fats, the waxes, the oils, and the steroids. Alright, so lipid, if we have to define what's a lipid, then it's no way to classify all the lipid together because there's nothing in them which is common. Right? So if we have to define a lipid, we define it as a diverse group of organic compounds which include fats, oils, hormones, certain components of membranes, and they are grouped together because they do not interact with water, right? So only thing that all of these lipids have in common that they don't interact with water. In short, we can say that all of the lipids, whether they are fats, whether they are waxes, oils or steroids, they are hydrophobic. They repel water. They do not interact with water. So another thing important is that these lipids are store of energy, right? They also used to store energy, but the difference between a carbohydrate and lipid is that carbohydrates is immediate source of energy, while lipids is not immediate source of energy, right? So largely our energy is stored in the form of lipids, while carbohydrates is immediate source of energy. So whenever you eat something, then immediate energy you get is because of the carbohydrates, while lipids they don't provide the energy on the immediate basis. Another similarity between lipids and carbohydrate is that both of these contain the elements of carbon, hydrogen and oxygen, but the difference is that the ratio is not 1:2:1. Right? So this ratio of carbon, hydrogen and oxygen is very much variable between these classes of lipids themselves, right? So they are insoluble in water. All of the lipids are insoluble in water. They are hydrophobic, they do not dissolve in water. You can carry out a simple test, just place a drop of water in an oil and you will say that water does not interact with oil. So both of these form separate layers, right? So lipids, whether they are fats, whether they are waxes, oils or steroids, they do not interact with water, they are insoluble in water, right? Likewise, I said that lipids are the macromolecules. It means these are polymers. So if these are polymers, what should be the monomer? So the monomer or the building blocks of majority of the lipids are glycerol and fatty acids, right? So the major building blocks for these lipids are glycerol and long chain of fatty acids. But since the lipids are of different classes like fats, oils, waxes, so each of these classes has different building blocks, or each of these classes are polymers of different type of monomers. But majority of the cases, if you have to say that in majority of the cases of lipids, the monomers are the glycerol and the fatty acid. So just for example, so I'm showing here the long chain of fatty acids. So what are fatty acids? Fatty acids are basically carboxylic acids. You can say here the carboxylic acid group here, and this is the alkyl group. But the difference between a simple carboxylic acid and a fatty acid is that fatty acids have long chain of hydrocarbon. Right, so these are the long chain of carboxylic acids. That's why they are known as fatty acids. So fatty acids may be saturated or unsaturated. So there may be a double bond in a saturated, or if there is no double bond in the fatty acid chain, then that's called saturated fatty acid. Right, so these unsaturated fatty acids, this double bond may be cis, such as shown here, or it may also be trans. Right, so saturated fatty acids or the long chain of fatty acid, while unsaturated fatty acids have some unsaturation. They may have presence of a double bond, and this double bond may be cis, and that double bond maybe trans. So you might have heard on the advertisement that say this oil is virtually trans fat free, right? So when the advertisement say this oil is virtually trans fat free, it means there is no fatty acid with this trans double bond. So all double bonds are either cis or there is some saturation there, right? So these are the trans fats. So you might have studied in the earlier semesters of biochemistry that why trans fat is not good for us, because if the oil has trans fat in it, it means it has a double bond which is trans, then such a double bond can be easily oxidized, right? So if a double bond can be easily oxidized, the result in formation of products which are not good for us, right? So that's why the oils or the companies which manufacture oil or they market oil, they always state that this oil is virtually trans fat free. It means apparently there is no double bond in the structure, right? So the unsaturated fatty acids, they may be cis or they may be trans. So if you have to classify the lipids, so as I mentioned earlier that they are very diverse class of organic macromolecules, right? So we can classify them in basically three categories: the simple lipids, complex lipids, and derived lipids. Among simple lipids, we have fats and oils and waxes. So these are normally the liquids, and these are the semi-solid material. And then we have lipids. So complex lipids. So among complex lipids, we have phospholipid, glycolipids, and lipoproteins. Right? So why we call them as complex lipids? Because they have a lipid component and some other component. For example, if a lipid is attached with a phosphate group, then such lipids are known as phospholipids. If a lipid is forming some complex macromolecule, or if a lipid is linked with a sugar group, then such lipids are known as glycolipids. And if the lipid is linked with some protein, then in that case the lipids are known as lipoproteins, right? So these complex lipids are also known as the conjugates, right? Because we have conjugated or we have attached a lipid with a phosphate group and a lipid with a sugar group or a lipid with a protein, right? So all of these are known as the conjugates, right? Lastly, we have the derived lipids. What are derived lipids? These include steroids and these include the terpenes. So as mentioned earlier, we will not go in much detail about their structure unless it becomes necessary. So you should know about the classification of lipids, and you should always remember that lipids are a very diverse class of compounds. So normally there is not much similarity in the structures of the lipids, and what makes them similar is just their interaction with water. So all of these lipids have one thing in common, and that thing is that all of these are water repelling. They are hydrophobic. So examples of lipids are many. Some lipids are important part of biological membranes. For example, phospholipids, you might have heard of the term. So phospholipid is an integral part of the biological membrane. And if you remember that these phospholipids contain the hydrophilic heads, so these heads are hydrophilic, right? And they have hydrophobic tail. So these tails are hydrophobic, right? So these tails are basically the lipids, and therefore you call them as lipid bilayer, right? So these tails are basically the lipids, so we call them as lipid bilayer, right? And also the lipids exist as waterproof covering, such as plasma membrane of cell and cuticle of plants. So you might have observed some oily type layer on the surface of leaves. So that oily type surface or covering on the surface of leaves, this is basically also the lipid. So you can just generalize that anything which is water repelling will be a lipid, right? So next we'll consider about the proteins. So proteins are, you might have heard, they are used to form bones, and they are used to form muscles, and they are an important part of the transport substances which transport material into and out of cell. They are also an important part of our immune system, and they help fight diseases. Also, proteins are important in controlling the rate of chemical reactions, and they also regulate the cell processes. So proteins are very much central in the medicinal chemistry, right? So we'll consider as we proceed further in this course, but you should consider the proteins in much more detail, right? So these are importance of proteins because they're important they are performing very much important functions in a biological system. So if you consider the elements which form proteins, the proteins involve the elements of carbon, hydrogen, oxygen, nitrogen, and sulfur. So again, the proteins are macromolecules. It means they are polymers. So since they are polymers, it means they should have some monomers, right? And the monomers, or the building blocks of proteins, are known as the amino acids. Right, so what's amino acids? It's a general structure I've shown here. So it has an amino group and a carboxylic acid group. That's why it's called amino acid. Right, so we have on one end we have an amino group, NH2 group, and on the other hand we have a carboxylic acid group. Right, so this is the common thing in all of the amino acids. So there must be carbon with a hydrogen here, and to this carbon should be a carboxylic acid attached and an amino group attached. So all of this fragment will be same in all of amino acids. So what is the thing that's making it different is the R group here. So just variant is the R groups. Just keep on varying the R group and we can have different type of amino acids. So all amino acids that are occurring in a biological system, all of them are alpha amino acid. Right, so we call them as alpha because both of these, amino group and the carboxylic group, are attached to this alpha carbon. So this carbon is important. This carbon is known as the alpha carbon. Right, so these amino acids are basically the monomers of proteins. So when they combine together, we get different type of proteins. So it becomes important for us to classify the amino acid because as mentioned in the previous slide, I said that this R group is a variant. So depending on nature of this R group, the amino acids are different types. So amino acids may be neutral amino acid. So in case if an amino acid is neutral, it means there must be one carboxylic group and one amino group. Right, so amino group, as you know from your basic chemistry, this amino group is basic and the carboxylic group is acidic. So since you have equal number of basic and acidic groups, so they neutralize each other. So that's why the amino acid in which there is in one amino group and one carboxylic group only, such amino acids are known as neutral amino acid. Right, so for example, you may have glycine, and I suppose you know that all amino acids are abbreviated by 3 alphabets. So this is GLY for glycine. Then we have alanine, valine, leucine, and isoleucine. So all of these are examples of neutral amino acid. Then we have another class of amino acid that's known as the acidic amino acid. So you can just predict when one carboxylic group is extra. So it means that if you have one amino group and more than one carboxylic acid group, then in that case the amino acid will be acidic amino acid. For example, aspartic acid, asparagine, glutamic acid, and glutamine. So all of these have extra carboxylic acid group. So you can see on the next slide when I'll show you structure. So you can predict which of these is acidic amino acid or which of these is basic amino acid. Right, so all of these are examples of acidic amino acid. Then we have basic amino acid. You can again predict that if you have extra amino group, then in that case the amino acid will be basic amino acid. So examples of basic amino acids are arginine and lysine. Then we have another class of amino acid which are sulfur containing. Right, so amino acid which have sulfur in their group, such amino acids are called sulfur containing amino acids. So we have two only naturally occurring amino acids which are sulfur containing. So these include the cysteine and methionine. Right, then we have alcoholic amino acid which have an alcoholic group. So these include the serine, the threonine, and tyrosine. Then we have aromatic amino acid which have aromatic ring. Then among these we have again two examples of phenylalanine and tryptophan. In the last category of amino acid are the heterocyclic amino acid, in which there is a heterocyclic ring containing a nitrogen. right? So in this category we have two amino acid. So you might have heard about this term histidine. So histidine and proline, so both of these amino acids are heterocyclic amino acids. Just let's have a quick look on the structures of these amino acids. So you can see here, so there's carboxylic acid and H2, so both neutralize each other, and here we have an extra carboxylic acid group. So you can predict this is an acidic amino acid. Again, let's see here, so again these are neutralizing each other, but here we have an H2 extra. So this NH2 is basic, therefore we can say that this lysine is a basic amino acid. So here you can imagine, here you can see an aromatic ring, so that's an aromatic amino acid. Here you can see again an extra carboxylic acid group, right? So you can predict this is a carboxylic, or this is an acidic amino acid. Here you can see these two are neutralizing each other, and here we have 3 nitrogens. So each nitrogen is basic, so this is highly basic amino acid. And here you can see this is histidine, and this histidine has a heterocyclic ring with nitrogen, right? So this is heterocyclic amino acid. So if you have a structure of amino acid, you can just see and tell whether that's acidic, that's basic, that's a alcoholic amino acid, or that's sulfur containing amino acid, or that's neutral, etc. So just by seeing the structure you can just tell which type of amino acid it is. Alright, so let's see that how the protein is formed. So as mentioned earlier, the proteins are the polymers of amino acids. So amino acids are basically monomers. It means that when monomers combine together, they form that polymer, right? And this is something very much important in case of the amino acid, that when these amino acids combine together, we say the amino acid condense. Actually, it's a simple reaction of organic chemistry. So carboxylic acid group reacts with the amino group and form amide. Right? You can just visualize this functional group. So this carbonyl and this is nitrogen. So this function is known as the amide group. Right? So in case of proteins, we call this amide function group as the peptide linkage. Right? So this bond between this carbonyl carbon and this nitrogen, this bond is known as the peptide linkage. Right? So since that's a condensation reaction, so you can say that when two units combine with elimination of water to form a larger molecule, so this reaction is condensation reaction. So this is the condensation reaction of two amino groups. So one will react with this carboxylic end and other will react with the nitrogen end. Right? So reaction of this carboxylic acid with amino group result in the formation of this peptide linkage, and this peptide linkage will carry on reacting. So this end at which we have a carboxylic acid group, this end is called C-terminus of protein. Right? So this end at which you have a carboxylic group, this end is known as the C-terminus, and the terminal at which you have an amino group, so this terminal is known as the N-terminus. Not terminal, right? This is terminus. So you have a C-terminus and an N-terminus in a protein. Right? So if you have a chain of two amino acids which are linked by peptide bond, such chain is known as dipeptide. Right? And if you have a large chain containing large number of amino acids, then such a chain is known as polypeptide, and polypeptides are also known as proteins. Right? So you can say that protein is basically a polypeptide. Right? So the sequence in which amino acids are present in a polypeptide is very much important, because if it changes sequence, the functioning of the protein becomes different. Right? So for a particular protein, the sequence of that amino acid is specific. So if you change the sequence, the nature of protein changes. Just you might have studied in the earlier classes about the sickle cell anemia. So in sickle cell anemia, what happens that one of the amino acid is changed, or the sequence of amino acid in a protein is changed. Because of that, the red blood cell changes its structure. Right? And because of the change in structure, the person having the defected amino acid faces the problem of breathing. Right? The sequence of amino acid is for very much important, and the sequence of amino acid in a polypeptide is specific for a particular protein. Then, if you have to classify the proteins on the basis of functions they are performing, then there are different types of proteins on the basis of their functional classification. So we have structural proteins such as keratin and collagen. Then we have enzymatic proteins such as pepsin. We have transport proteins like hemoglobin. Then we have hormonal proteins such as insulin and growth hormones. We have contractile proteins which are responsible for contraction of relaxation of muscles. So these proteins include the actin and myosin. Then we have storage proteins such as oval albumin and hemp. Then we have genetic proteins which are known as nucleoproteins. Then we have the defense proteins which are important in our immune system, and these are known as immunoglobulins. And then we have receptor proteins. So among receptor proteins we have dopamine receptor and insulin receptor, etc. Right, so in case if you have to classify the proteins on the basis of their structure, right, so in that cases, so you might have studied about the primary, secondary and tertiary structures of the protein. So this is linked with that. So proteins are basically divided into three groups on the basis of their complexity. So there are simple proteins, conjugated proteins and derived proteins. So firstly we'll consider the simple proteins. Simple proteins are further divided into two parts. These are the globular proteins and fibrous proteins. So among globular proteins we have albumin, globulin, histones. Then among fibrous proteins we have collagen, elastin and keratin. Right? So conjugated proteins, in that case the protein is linked with some other macromolecule. For example, when a protein is linked with a nucleic acid, then such conjugated proteins are known as nucleoproteins. If a protein is linked with some sugar group or some carbohydrate, then such proteins are known as glycoproteins. Right? Similarly, we have mucoproteins, we have lipoproteins, we have phosphoproteins, we have chromoproteins and metalloproteins. Then we have derived proteins. So derived proteins are further divided into categories. These are the primary proteins and the secondary proteins. Among primary we have coagulated proteins and proteins and metaproteins. Then among secondary we have proteases, pentoses and peptides, etc. So we will not go in much detail about that, assuming that you already know about these type of proteins. Right? So proteins are found in large number of foods, such as it's found in red meat and chicken and fish and eggs and these cereals and pulses, etc. So next we will consider the class of macromolecules which are known as the nucleic acids. Right, so you might have heard about the term that nucleic acid transfer, transmit or store genetic or hereditary information. So these are the molecules which are responsible for our genetic makeup. Right? So the elemental composition of these nucleic acids is carbon, oxygen, nitrogen and phosphorus. Right? So just compare that in case of the carbohydrates we had only three types of elements. There were carbon, hydrogen and oxygen. Lipids also had the same elements of carbon, hydrogen and oxygen, but the ratio is different from that of carbohydrates. Among proteins we had carbon, hydrogen, oxygen, nitrogen and sulfur, and in nucleic acid in place of sulfur we have phosphorus. Right? So nucleic acids are of basically two types, the DNA and the RNA. So since the nucleic acids are macromolecules, so it means they must have some monomers or the building blocks. So these are polymers, and nucleic acids are polymers of nucleotides. So the monomers of nucleic acid or the building blocks of nucleic acids are known as the nucleotides. So let's consider a structure of a nucleotide. So this is the monomer of nucleic acids known as the nucleotide. So you can say here that it has the three components. So one of the component of a nucleotide is a sugar group. Right, so this is a sugar, and here we have a nitrogenous base, and here we have a phosphate group. Right, so phosphate group, a sugar and a nitrogenous base, these together form a nucleotide. So the structure is very much specific if you consider in detail. So only the sugar should only be a 5-carbon sugar. Right, so you can see that this sugar is in the form of a cyclic form. So this sugar has 5 carbons. So that's a pentose sugar. So in nucleic acids, the sugar is always a pentose sugar, right? So also it is very specific that this nitrogenous base will always attach on this carbon, right? So this carbon to which this nitrogen is attached, so suppose if you have to number that, so this oxygen is given number 1 and this carbon is given number 2. So nitrogenous base is always attached on carbon number 2 of the sugar group, right? And the phosphate group is attached, so if this carbon number 2, 3, 4, 5, 6. So this carbon, this is the 6th carbon that has always the phosphate group attached to it, right? So this position of these groups can never change in nature. So in nature, a nucleotide always have a nitrogenous base on this carbon and a phosphate group on this carbon, right? So what makes the DNA and RNA different is just this group, just this hydroxyl at carbon number 3, right? So you can say that if you have to number that, so you have to number oxygen as first, then carbon as second, and this third. So this hydroxyl at this 3rd carbon, right? So in case if this hydroxyl is present, then we have, this is RNA, right? This is ribose nucleic acid. But in case of DNA, there is no hydroxyl group, so that's why we call as deoxy. Oxy means oxygen and D means no or eliminated. So if you remove this oxygen from here, then the sugar will be deoxyribose. And if you have this hydroxyl here, then sugar will be ribose sugar, right? So in case of DNA, what's the difference is that sugar. So in that sugar, this hydroxyl group is missing, right? So that's why the DNA stands for deoxyribose nucleic acid, right? Also, these nitrogenous bases may be different. So these nitrogenous bases are of 2 types, the purine bases and the pyrimidine bases. So change in the purine or pyrimidine bases result in different type of nucleotides, right? So DNA has 4 different nucleotides that include the adenine, guanine, cytosine, and uracil. Cytosine and thymine. So in case of the DNA, the nucleotides, the 4 different nucleotides are based on the difference in this nitrogenous base. So these nitrogenous bases may be adenine, guanine, cytosine and thymine in case of the DNA, and there should be no hydroxyl here. But in case of the RNA, what happens that this hydroxyl is here and the bases may be adenine, guanine, cytosine and uracil. So instead of thymine, which is present in DNA, the RNA has uracil. So this is the basic difference in the structure of the DNA and RNA. So here we can see the structure of DNA and here we can see the structure of RNA. So DNA is always double-stranded structure, right here, and this is the single structure, and the structure is of what you say as RNA. So this is ribonucleic acid, right? So you can see that in ribonucleic acid, this is the hydroxyl group which is retained, right? In case of RNA we have here uracil, but in case of DNA we have all the 3 are same, but instead of uracil we have thymine here, right? So this is a brief summary about the nucleic acids. So next we will consider about the enzymes. So enzymes are basically proteinaceous substances, means they are made up of proteins, and the enzymes are produced by the living systems. And the production of these enzymes is controlled by genes, right? And what are genes? Genes are fragment of DNA, right? So here you can think of a link between the proteins and the DNA and the genes. So basically enzymes are proteinaceous substances, means that are made up of proteins, and these are made up of proteins under the control of enzyme. So this protein production is controlled by enzyme, and the enzymes, these are controlled by the genes, and genes are basically small fragments of DNA. So DNA is basically controlling the production of these proteinaceous substances. And what's specific about the proteins is that they are known as the biological catalyst. Right? So if you have to define a catalyst, what happens? The catalyst is any species that speeds up a reaction and is recovered unchanged by the end of reaction. Right? Same applies to catalyst, and the same applies to enzymes as well. The only difference between enzymes and catalysts is that the enzymes are biological catalysts. They are only found in the living system. Right? So enzymes here, they enhance the rate of reaction. In the branch of science which deals with the study of enzymes, their nomenclature, their reactions and their functions, this branch of science is known as enzymology. Right? So enzymes occur in colloidal state. Right? They are normally found in the form of a suspension. Right? So they are normally occur in the colloidal state and are produced in inactive form. The inactive form of this enzyme is known as the proenzyme, and the proenzymes are normally known as zymogen. Right? So if an enzyme is produced in inactive form, the inactive form of that enzyme is known as zymogen or proenzyme. So this zymogen is converted into its active form by specific factors like a pH or presence of a substrate as well. Right? So enzymes are present always at all times inside your body, but when there is specific change in pH, then that enzyme which is acting or existing as zymogen, then this enzyme is being converted into its active form. Right? So enzymes are of two types. The enzymes that are produced within a cell for metabolic activity. So enzymes that are produced within the cell, they are known as the endoenzymes. And the enzymes which are produced inside the cell but they act away from the site of their synthesis are known as the exoenzymes. Right? So the difference between endoenzyme and exoenzymes is that the endoenzymes are produced in the cell and perform their function within the cell as well, while exoenzymes, they are produced in the cell but they act away from the site of their synthesis, so they are known as the exoenzymes. So these terms will appear later when you study the course of medicinal chemistry. So you should know about what do you mean by the endoenzyme and the exoenzyme. So now we'll have the general properties of enzymes and the factors which affect their activity. You should know certain things like the enzymes accelerate the reaction, but they cannot initiate a reaction. Right? They make the reaction to work faster, but they cannot cause the reaction to occur. Right? So enzymes accelerate the reaction, but they do not initiate them. Second, the enzymes do not participate in the reaction and remain unchanged at the end of reaction. Right? So since they are recovered by the end of reaction unchanged, so therefore they are needed in very small quantity. Right? So the second important thing you should know is that the molecule of enzyme is very much larger than the substrate molecule. Right? So the molecule of the enzyme is very much larger than your reactant, and only a specific part of the enzyme comes in contact with your reactant molecule. Right? And that specific part of the enzyme which comes in contact with your reactant substrate is called the active site. So I repeat that enzyme is a very large molecule, right? It has a very large surface area, and the reactant which has to be reacted with the enzyme, this substrate molecule comes in contact with only small part of that enzyme. Right? And this small specific part of the enzyme which comes in contact with the substrate is known as the active site. Right? Secondly, these enzymes are of amphoteric nature, means that they can react with acidic substances as well as alkaline substances. Why is that so? Because the enzymes are proteins, and you know that proteins are made up of amino acid. An amino acid has an amino group as well as carboxylic acid group as well. Right? So because they have acidic group as well as basic groups, therefore they can interact with acidic substances as well as the alkaline substances or basic substances. Therefore we say that enzymes are of amphoteric nature. Enzymes are specific in nature, so a single enzyme will act upon a single substrate or group of closely related substrate. For example, if an enzyme works on a carboxylic acid, then this enzyme will only work on carboxylic acid, right? Whether you can change the R group of carboxylic acid or not. So enzymes are very specific in their action. A specific enzyme, a single enzyme will act on a single substrate, single subtype of substrate, or a group of closely related substrate. For example, just another example, the enzyme urease. This enzyme only acts on urea. Similarly, the invertase enzyme only acts on sucrose and no other carbohydrate as well. So the configuration of a substrate molecule, if different, right? If the configuration of substrate molecule is different, then different enzymes will be required. For example, if you have substrate with cis double bond, that is being reacted with the enzyme, and if you change the configuration from cis to trans, the same enzyme will not work. You have to make use of a different enzyme. Similarly, if the configuration of a substrate is R, then a specific enzyme is useful for R, but if you change the configuration of your substrate to S, then you have to require a different enzyme. So this is the specificity of enzyme. They work on a single type of substrate, and they're also the configuration specific. So if you change the structure of your substrate, then the enzyme will no longer work on it. The sixth property of enzyme is that they are colloidal in nature, right? And because of their colloidal nature, right, their gel-like nature, they provide large surface area for reaction, right? So this is the advantage of having a colloidal nature. And these colloids are a mixture of two components, right? So enzymes, since it is occurring in the form of colloidal nature, right, it's existing in the form of like colloid gel-like material, right? So if it's existing in the form of gel-like material, it means it has two components, right? So one component is the dispersed particles, and the second component is the dispersion medium, right? So the enzymes consist of two components, the dispersed particles and the dispersion medium, right? And it's also important for you to note that the size of the dispersed particle is always larger than the dispersion medium, right? The dispersion medium, which is the gel, right, it has smaller size, while the particles which are suspended in this medium, these particles are of larger nature. Then we have enzymes optima. So enzymes generally work best under certain narrowly defined conditions. So these narrowly defined conditions under which enzymes work the best are known as the optima, right? So these conditions include the temperature, the pH, the concentration of substrate as well. So we'll see these one by one. So firstly, the temperature sensitivity. So since you know that enzymes are proteins, so since proteins are affected by temperature, similarly enzymes are also affected by temperature, right? So there is a certain range in which enzymes work the best. So with increase in temperature, the activity of enzyme increases up to 40 degrees C. However, if you increase the temperature above 60, the proteins undergo denaturation or even complete breakdown, right? So enzymes can't work well above 60 degrees C, right? However, when you reduce the temperature to freezing point or below freezing point, the enzymes become inactivated, but they are not destroyed, right? So the rate of reaction is maximum at the optimum temperature, and the optimum temperature, that's the best temperature for which the enzymes to act, is about 40 degrees C. So you can see if you increase the temperature above 60, you destroy the enzymes, but if you freeze the enzyme, in that case you inactivate the enzyme, but they are not destroyed. So if you increase the temperature of those frozen enzymes, then around 40 degrees C, those enzymes will start to work again. Then you have pH sensitivity, right? Most of enzymes are specific to pH and remain active within that particular pH range, right? And if you use strong acid or strong base, then enzymes will be denatured, right? So most of the intracellular enzymes, the endoenzymes which are called, so most of these enzymes work the best around a neutral pH. Certain enzymes work under mildly acidic conditions, certain work under mildly basic conditions, but most of the enzymes work under normal neutral pH. Then we have next factor, which is concentration of enzyme in the substrate. So when the concentration of reacting molecules is increased, the rate of reaction increases, right? So the enzyme reaction increases up to a certain limit, but if you keep on increasing the concentration of enzyme, then we say that enzyme becomes saturated with the substrate and the activity becomes steady, right? So if you increase the substrate concentration, the rate of reaction goes on increasing, but beyond a certain concentration, the enzyme becomes saturated because it can no longer attach any more substrate with it, and the activity becomes steady. So there is no decline in the enzymatic reactions. The activity either increases or either remains constant. So when you increase the concentration beyond a certain limit, then activity of enzyme becomes constant. It becomes steady, it cannot decrease. The ninth factor is the enzyme inhibitors. These are certain products or certain material which inhibit or stop the enzyme activity. So if you somehow block the active site of the enzyme, then the activity of the enzyme is lost, right? The activity of enzyme is because of this active site, and if you somehow block this active site, then the enzyme activity is lost. So the material or the products which inhibit the enzyme activity by blocking the active site, such materials are known as inhibitors, right? So these substances have similar structure to the substrate molecule, right? The inhibitors must have structure similar to substrate molecule because, you know, as mentioned earlier, that enzymes are substrate specific. So they work on specific type of substrate or related substrate. So if you have to block the active site, you have to make use of a thing which is similar to the substrate with which the enzymes react, right? So these inhibitors, right, these are substances which block the active site of the enzymes and result in loss of activity of enzymes, right? So here we have classification of enzymes, right? So depending on the type of reaction they catalyze, the enzymes are grouped into different categories. So we have oxidoreductase enzyme. So these reactions which catalyze is that they transfer oxygen or hydrogen atoms, or they transfer electrons from one substrate to another, right? Example, dehydrogenase enzymes or oxidase enzyme. Then we have transferase enzyme. So transferase enzyme transfer specific functional group from one substrate to another. For example, if you transfer amino group from one substrate to another, then in that case the enzyme will be transaminase. Trans means it's transferring, and aminase means amino group. Then hydrolase enzyme, they hydrolyze a substance. For example, the digestive enzyme, since they hydrolyze the material, therefore they are known as hydrolases. So isomerase enzyme, as the name suggests, they causes isomerization. For example, phosphohexoisomerase. Lysase enzyme, these are non-hydrolytic. They don't hydrolyze, right? So what they do is that they remove or add a group to a substance, but they don't cause the hydrolysis. For example, decarboxylase enzyme. So these will cause the removal of carboxylic acid group, but they will not cause any hydrolysis. Similarly, aldolase enzyme. So here we have ligase enzyme, right? So ligase enzyme are used for joining two molecules together. So these result in formation of new bonds. For example, citric acid synthetase. So this enzyme is useful for joining the two molecules together. So that's all for the lecture of introductory lecture of biomolecules. So here's your assignment. So you have to differentiate between simple and complex sugars. You have to name some simple and complex sugars as well. Then you have to compare the lipids and carbohydrates. Also, you have to tell, if you are starving, which of these biomolecules will you prefer, right? And then you have to tell me about the difference in DNA and RNA and what are the factors that affect the enzyme activity. So if you're looking for the recommended books, here are the 2 recommended books you can look for. Thank you and good luck.